All right, so here's an overview of Carb Lecture 4. We're going to talk about glycogenesis and glycogenolysis and their regulation. And then we're going to talk about gluconeogenesis and how it's regulated. And then we're going to talk about the Krebs cycle and ETC. And then we'll kind of put it all together about, and this will include how ETC works and how things like oxygen deprivation change substrate to carbohydrate instead of also fat. So some of this is review. We start off like we've seen the structure of glycogen before. We know glycogen is all glucose. It's the storage form of glucose in animals. And we know that we store it where? Muscle and liver, two main tissues that have glycogen for us. We know it's alpha 1 to 6, alpha 1 to 4 bonds. And it's got a significant amount of branching, and that allows a high number of glucose molecules to be stored in a smaller volume. So it's a pretty dense molecule. You can actually see it if you isolate liver cells, for example. You can look at our light microscope, and if you have your conditions just right, you can actually see like little glycogen crystals in there. And it's in the cytosol. And then you could see, like in a muscle, it's a little bit harder to see, but you can look at, before and after exercise, you can actually see whether or not there's a visual glycogen. So there can be quite a bit of it. Remember, we talked about that. In the liver can be 4 to 8%. And one of the other tricks about glycogen is it tends to attract water. It takes about 4 grams of water to store each gram of glycogen. It tends to accumulate. So if a person's lost significant glycogen and deplete their muscles, that's another reason why they'll lose weight, because they'll have less water in a muscle fiber. And so what that means, though, is that high branching means high energy. The enzyme that breaks off glucose works on the non-reducing ends. So the more branching you have, the more ends you have. And we'll talk about this in a second, but imagine an enzyme next to every one of these filled in circles. So when that enzyme is activated, you can get significant amounts of glucose in a very short period of time. And where would that enzyme be? In the liver and muscle. So we'll talk about its regulation. So here's glycogenolysis. I mentioned this before, right? It's got lots of glucose units. In humans, the liver is completely depleted of glycogen by 18 to 24 hours of fasting. Remember that? That's kind of where we go from fasting to starvation. When that glycogen is gone in the liver, our metabolism starts to significantly change, because now we've lost a key fuel source for a lot of tissues in the body. So we'll talk about ketogenesis in a week or two, but that's when that really starts changing, where ketone production goes up. Our body senses that depletion and responds by looking for an alternative energy source. So that's a key point. All right, so the enzymes involved in breaking down glycogen, there's 2 of them. One is glycogen phosphorylase. That's the major one for our class. This one breaks alpha 1 to 4 bonds. It uses a phosphate to do that. That's why it's called phosphorylase. And I'll show you that in a second. There's another enzyme called a debranching enzyme. That's the one that breaks the alpha 1 to 6s. And this is actually a really interesting enzyme, because it breaks the bond and then it moves a chain of glucose molecules, makes an alpha 1 to 4, so the glycogen phosphorylase can actually work on it. Fascinating. We don't have time to talk about it. Okay? So it's a multifunctional enzyme and not highly regulated. So this is why we're going to focus on glycogen phosphorylase. This image is in your textbook, and I have it up here to show the key reaction here. Two important reactions, actually. So imagine we've got a big glycogen molecule. Now we're looking at the end of one of them. So imagine this one is one of those black circles, the filled in circle. And you can see it's non-reducing end. Why is it non-reducing? Its anomeric carbon is in the glycosidic bond. So we're on the end of it. This enzyme, glycogen phosphorylase, is going to hydrolyze right here. not hydrolyze, sorry. It's going to break this bond. This is a covalent bond. Covalent bond, in order to break it, you got to add electrons. Where do the electrons come from? Phosphate. So the enzyme uses inorganic phosphate, takes those electrons and puts it here so that this glycosidic bond is broken by adding a phosphate to this oxygen. So you end up with a phosphate right here. And because it adds it, that's carbon number 1 that has that hydroxyl group in the phosphate, we call that glucose 1-phosphate. That's the major product of glycogen phosphorylase. And again, now if we go back to that glycolysis diagram, you can see, oh, now we've got glucose 1-phosphate, we can go to glucose 6-phosphate. Right? And then where can glucose 6-phosphate go? Glycolysis. So we can go glycogen to glycolysis. Right? That makes sense, right? When would we be breaking down glycogen? Fasting. Anytime our blood glucose starts to drop, hepatic glycogenolysis is elevated. And some of that glucose from the glycogen goes through glycolysis. Why? Because we're fasting. We probably need to produce more energy. It's amazing how often all this stuff happens every day, right? And you want to change this? Eat something. You want to change it again? Don't eat something. Just always happening like this. And I think a lot of people don't, we realize this, but a lot of people don't. Like, I don't know, I'm tired all the time. How's your nutrition? Because it affects hormones that affect how we feel. And so we have to continually kind of eat what we need. And this is one of the things that happens all the time because glucose is so important. Okay? Now, here's another option. The liver can do this. Go glucose 6 to glucose. Now, if you're going to name this enzyme, it's like a game show. Name this enzyme. Right? A lot of times we want to start with a substrate and then add what it does. Glucose 6 phosphatase. Ding, ding, ding. Right? Phosphate's coming off. Now what do you have? Glucose. Now what can happen to the glucose? Out of the cell. All right, so that's two key potential destinations for the glucose from glycogen. Glycolysis or the blood. Please hold your applause when you see the complexity of this next slide. Right? I don't remember why I was so desperate. I'm like, I don't see a diagram for this. I'm going to write it on a piece of paper. And of course, I made a mistake on it, but I'll show you what that is. So here's what we're looking at. Here's glycogen phosphorylase making this. And then we said we can do this. We just said in the liver we can do this. The liver has glucose 6 phosphatase. Muscle does not. That's very important. Muscle does not express glucose 6 phosphatase. So in muscle, when we have glycogenolysis, it's going for glycolysis. In the liver, glycogenolysis can lead to glucose going into the blood or glycolysis. So that's what I'm missing on here. I should probably have put glycolysis for liver too. All right, we mentioned that. But anyway, that's an important point of this. Yep.
How is it going from glucose 1-phosphate to 6-phosphate?
Here?
Yeah.
That's an isomerase that just moves that phosphate.
Okay.
Yeah, we don't have to know that enzyme. Yeah, pretty easy conversion. Good question. And you're going to love this next slide or not like it at all, okay? Regulation of glycogen phosphorylase. And I would encourage you to think about this big picture so that you understand it and then use this slide to kind of support that. If you just memorize this slide, you won't understand how it works. So this can help you, but there's more to it than this. So this is in your textbook, and I modified it a little bit. But the key to this is the reaction we're talking about. Glycogen to glucose 1-phosphate, all right? So if you're going to add a word to this is phosphorylase. More specifically, this is glycogen phosphorylase. So if you want on here, you can get rid of that A. I don't care about that. This is glycogen phosphorylase. Glycogen phosphorylase is a, I wouldn't say a tricky enzyme, but it's a very complex enzyme that has lots of different structures. It's got an active form, a more active form, a very active form, a less active, and it changes activity based on a few different things. Okay? So let's just focus on the lower part of this. How do we go from an inactive form to an active form? Covalent regulation. And what's the most common covalent regulation we talked about? Phosphorylation. So this is phosphorylation. So in other words, let's say this is glycogen. And this enzyme's basically pulling glucose off and making glucose 1-phosphate. This enzyme has a phosphorylation site. So what goes into that? An enzyme uses ATP, pulls the phosphate off, adds it to a phosphorylation site on glycogen phosphorylase. So this is an enzyme. Name this enzyme. And you don't have to remember this one, but just so you know what it's doing. We don't have to remember. Glycogen phosphorylase. Sorry, phosphorylase. I'm making up enzymes now. Glycogen phosphorylase kinase. Adds a phosphate to this. That's what's happening here. Glycogen phosphorylase kinase activity is regulated by epinephrine and glucagon. So let's take a step back and go, well, where's epinephrine? When is it elevated? Stress, including physical activity. Epinephrine goes up. Epinephrine goes up, it stimulates glycogen phosphorylase by activating glycogen phosphorylase kinase. So you can just say epinephrine stimulates glycogen phosphorylase activity. That's fine. And does that make sense? It does, because during physical activity, some glycogen can be broken down to provide glucose for fuel. Right? We can go glucose 1 to glucose 6 to glycolysis. So let's say we start exercising. And in general, the higher the intensity exercise, the more epinephrine is released. So the more we stimulate glycogenolysis. And we're stimulating it through glycogen phosphorylase activity. So epinephrine stimulates that in muscle. That makes sense? So muscle fibers have epinephrine receptors. Now, the other thing that epinephrine does is it provides fuel. So another way to do this is epinephrine also stimulates activity of glycogen phosphorylase in the liver. So next to epinephrine, put liver. Okay? Glucagon. What's the main function of glucagon? To raise blood glucose. Which tissue will do that? The liver. So glucagon goes up. Its main function, raise blood glucose. One way, stimulate glycogenolysis in the liver. So we can go glucose 1-phosphate, glucose 6-phosphate, glucose to the blood. You can look at the previous diagram to look at that, right? This glucose can end up in the blood. Main function of glucagon, raise blood glucose. Do muscle fibers have glucagon receptors? Why or why not? Who says yes? Why not? Should muscle fibers have it? No, why? They won't release glucose. If this is in a muscle fiber, it would go glucose 1-phosphate, glucose 6-phosphate, glycolysis. So this doesn't happen in muscles. Glucagon does not stimulate muscle fibers because muscle fibers do not have glucagon receptors. So that one's just the liver. Everyone got that? Okay, so that's what's activating it. So if we want to inactivate this form of the enzyme, remove the phosphate. The enzyme that does that would be called glycogen phosphorylase phosphatase. You don't have to remember that, but that's what's happening. Another enzyme comes along and goes, erases that. Enzyme becomes less active. Okay? So these are transient upregulations. Another enzyme will come along later and go, signal's gone, decrease activity of that. Okay? Here's another form of the enzyme. So this is covalent regulation by hormones. Upper one, allosteric regulation. Again, systemic with the hormones, cellular by allosteric regulation. Can they work together? Yes. Can they be working at the same time? Yes. I think most things that you read will say hormonal is probably priority, but I won't ask you that. So now look what happens. See the way it says phosphorylase B? That's glycogen phosphorylase. This is a different but active form. And this form is obtained when molecules bind to its allosteric site. So for example, here's ATP, and what's up there? AMP, right? These are cellular molecules whose concentration is going to depend on energy status of the cell. So ATP, should it increase or decrease glycogenolysis? ATP goes up, glycogenolysis goes down. Don't need to break down more glycogen. Already have a lot of ATP. Okay, so when ATP binds that allosteric site, pushes it to an inactive form. Opposite of true is true for AMP. And ADP does as well, but AMP is more potent. So AMP is signaling for low energy status. It activates glycogen phosphorylase. Right? So they don't have it here, but what reaction could we put up at the top? This is active. We could put this reaction right here too, right? So once we're in an active form, we're going glycogen to glucose 1-phosphate. Everyone got that? It's a confusing slide or helpful? Okay, half and half, I'd say. So, like I said, just work through it and then that look back at the slide and say, oh, okay, that makes sense.
Did you say that AMP activates it or inactivates it?
Activates it. AMP activates it. Got it? So that's glycogenolysis. Now let's go to the opposite. Here's making glycogen. All right, remember the initial molecule is glucose. It's converted to, glucose enters the cell, goes to glucose 6-phosphate. And then after that, it's converted to glucose 1-phosphate. We'll look at the diagram in a second. And then there's 2 enzymes involved in glycogenesis. Again, we'll focus on one, and that's glycogen synthase. And glycogen synthase makes linear glycogen molecules. So what kind of bond can it make? Alpha 1 to 4. There's another enzyme called the branching enzyme that, again, will actually take pieces and move it and make branch points. So this one makes the alpha 1 to 6. All right, all this takes place in the cytosol. Glycogenesis is in the cytosol. You know that from week 1 or 2. Where does glycogenolysis occur in the cell? Cytosol. Okay, and this probably don't even have to say this. If glycogenolysis is upregulated, glycogenesis is downregulated. Right? In general, we're either trying to store glycogen or break it down for fuel. So here are some of the reactions. I think there's, I hope this is pretty clear. Again, this is in your text. So here's the synopsis. This happens in liver and muscle. They take glucose 6-phosphate, convert it to glucose 1, make an intermediate that involves a uracil diphosphate. And then they use that with glycogen synthase to make more glycogen. So here's the reaction. And this is actually simpler than it looks probably. Glucose, glucose 6, glucose 1. And here's a kind of weird reaction. This enzyme, its substrate is UDP. It kind of uses that as a binding point for the enzyme. So this is uracil diphosphate. So this would have a kind of a ring structure, nitrogen containing. So the enzyme uses that as its kind of binding point. So this is all that means. UTP is split. So 2 phosphates come off. And so essentially what you're adding is uridine monophosphate. So here's our uridine monophosphate. It was added to glucose 1-phosphate. So we just call this UDP glucose. Because now it's 2 phosphates. Uridine diphosphate glucose. That's what you usually call it. It should probably be more specific and say uridine diphosphate 1 glucose, but we don't call it that. Okay? So then this is used by, don't worry about glycogen and primer. This molecule is used by glycogen synthase to add glucose to glycogen. That's all that means. And we're going to talk about 2 ways it's regulated. So this is regulation of glycogen synthase. It's upregulated by insulin. That makes sense? Blood glucose goes up, insulin goes up, glycogen synthase is activated. That stimulates glycogen synthesis in liver and muscle. This enzyme is a little bit different. It's actually activated by dephosphorylation, the opposite of glycogen phosphorylase. So insulin causes dephosphorylation of the enzyme, activates it. There's a second thing that I want you to remember activates glycogen synthase. And that is glycogen depletion. So the two scenarios where glycogen synthase is activated, one is increased insulin. The second one, glycogen depletion. So this is one of the reasons why if someone is exercising, it's commonly beneficial if they consume carbohydrate immediately post-exercise. That carbohydrate will stimulate insulin release, activate the enzyme. That's also a point where glycogen levels tend to be depleted. So the enzyme activity is enhanced by both of those. And so that can allow the person to store more carbohydrate, specifically mostly in muscle, and that can help them in the next exercise bout. So typically post-exercise, as soon as possible, glucose should be consumed.
Does that only apply to people who work out normally, not someone who just does it unoccasionally?
So really good question. Our muscles actually get better both ways. The muscles can get better at breaking down glycogen. They can get better at storing glycogen. So it takes some training to kind of optimize that process. So the less consistent the exercise, the less important the carbohydrate intake is after exercise. Yeah, but if we want to maximize it, remember we said, if you do it right, you can double your muscle glycogen levels. That would be repeated bouts of exercise with repeated instances of carbohydrate intake. And that allows it to be accentuated. Yeah, so your muscles kind of learn how to do it better. Opposite of that, glucagon. Glucagon tends to downregulate glycogen synthase. Okay, let's talk about gluconeogenesis. So we said before that the purpose is to make glucose of non-carbohydrate precursors. And gluconeogenesis is mostly in the liver, probably at least 90%. Sometimes if starvation extends, the kidney cells start performing gluconeogenesis, but it's typically not very much. It provides a method to make glucose when intake or stores are low, and we'll look at non-carbohydrate sources in a minute. But can anybody name one? Amino acids. Glycerol. We'll look at that in a second. What about this one? And lactate. Remember Cori cycle? So if you want in your notes on this one, you can also put, so amino acids, glycerol, lactate. Those are the three most common. Of those, amino acids is the most significant. Right, remember, this is in the liver. This is a good diagram to summarize this. Left side of this pathway is glycolysis. We've seen that a few times. Right side, gluconeogenesis. It's essentially reverse of glycolysis. Now, one of the key points is some glycolysis reactions are unidirectional. This one is. Right, that enzyme, hexokinase or glucokinase, do this. They don't go backwards. We need a different enzyme. That's where glucose 6-phosphatase comes in. You can already see, oh, that's why muscle doesn't do gluconeogenesis. It doesn't have this enzyme. Gluconeogenesis is making glucose, not glucose 6-phosphate. Bidirectional. Same enzyme will run it either way. So it's not limiting for gluconeogenesis. So here's the next problem for gluconeogenesis. PFK1 runs it one way. You want to reverse that, you need a different enzyme. That enzyme is called fructose 1,6-bis phosphatase. Takes off the phosphate to get back to glucose, to get back to fructose 6, right? Bidirectional, same enzyme, bidirectional, 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 till you get to here. Phosphoenolpyruvate is converted to pyruvate by pyruvate kinase. Reverse of that is multiple steps. So pyruvate to PEP is what we call the pyruvate kinase bypass. So here's the point. Why doesn't every tissue perform gluconeogenesis? Because some tissues lack enzymes or mitochondria. All right, so we could say, well, muscles lack glucose 6-phosphatase, red blood cells lack mitochondria. All right, liver can match all of those requirements, so liver cells can do that. So what I would say is, and you'll be able to see on the study guide, I'll try to make it clear on there, that this is a good diagram to know. But what you can do is, this part, you can just put arrows. You won't have to name it. But you'll need to be able to plug in enzyme names. So don't forget pyruvate kinase is for glycolysis. Its reverse, pyruvate kinase bypass. You don't have to remember these. Uh-huh.
So we would only have to know the ones in the box. Right?
Yes, yeah, good. Yep.
The ones that are different cleavage.
Yes. Kind of boasts all that table right there. So this is another way of looking at it, and this kind of shows our substrate as well. Right, here's lactate, which reverses to pyruvate. But now we have pyruvate, so what do we have to do? Pyruvate kinase bypass. And this is a really good, I have this here because it's a good visual. Pyruvate has to go into the mitochondria. And there's enzymes in the mitochondria that allow it to go back to malate. Remember malate aspartate shuttle? There's a transporter here, so malate can go out. Once you have malate in the cytosol, remember, it can convert back to oxaloacetate. Right? And then the oxaloacetate can be used for malate aspartate shuttle. So there's multiple uses for that. And then once we have oxaloacetate in the cytosol, it goes to PEP. So now we can reverse. Got to have the mitochondria to do that. So in other words, if you think about it, Cori cycle generates lactate. We looked at that diagram. Lactate in the liver can go back to glucose. So lactate goes into the liver cell, becomes pyruvate, goes into the mitochondria, the PEP, and then back to glucose. There's not really anything easy about it. So again, it's another indicator. Glucose is really important. This requires energy to do that. Remember that from the Cori cycle? So gluconeogenesis requires energy. We're investing energy to get glucose. So that's one of our substrates. And then we have, what's the other one? Glycerol. Where's most of that glycerol come from? Triglycerides. So if we're fasting or starving, fat oxidation goes up. I guess that's kind of one of the good things, right? Oh, burning more fat. You are because you have to. What that does is now triglycerides, let's say in my fat tissue, the triglycerides are broken down to fatty acids and glycerol. Glycerol goes into the blood, goes to the liver, and the liver can take that glycerol and make glucose. So you get a little bit of glucose from triglycerides, not from the fatty acids, right? So we get a little bit of glucose from that, and that's kind of what that shows, right? 3 carbons goes DHAP back to glucose. Got it? Uh-huh.
So for the lactate,
Yep.
it goes to pyruvate and then does it go like should that be like a reversible arrow also to PEP?
Here?
Yeah.
No. Gotta go this way.
Oh, so that goes through also.
Yeah. Gotta go that way. Gotta bypass that. Right? So again, gluconeogenesis is always occurring, but guess which hormone elevates it? Glucagon. Glucagon increases gluconeogenesis. What does insulin do? Decreases gluconeogenesis. Again, if we just integration of these pathways, if glycolysis is up, gluconeogenesis is down. If gluconeogenesis is up, glycolysis is down. Right? All right, energy production. We saw this one before, and we said most energy from our food and basically our fuel sources comes from the electrons. It's about oxidizing those molecules that they can make. And the reason is because the electrons are used by ETC, and ETC is phenomenally effective at making ATP. Right, using those electrons. So we consume carbohydrate, fats, proteins, convert them to molecules that can be oxidized to provide electrons for ETC. And a big part of that is Krebs cycle. So you guys already know this, but regulation of Krebs and ETC are linked. And that's what we're going to talk about in this. So a couple key points, remember? I know this is like Khan Academy, is that what it's called? You guys, does anyone still see that? Everyone watch that? ETC down here, remember? So here's our oxygen. Gotta have oxygen to run ETC. Because as those electrons go through ETC, they gotta end up somewhere to take them out of that chain of events. So they end up combining with oxygen to make water and ATP. So let's talk about the regulation of this. So we're going to go through Krebs cycle. This is kind of what you need to know. I'll let you look at this later. So let's go straight to this. This is the level of detail that I want you to be able to draw. How many of you guys have drawn the Krebs cycle before? Okay. So hopefully at least half of you. So this should be review. So again, we're in the mitochondrial matrix. Krebs cycle starts when acetyl-CoA condenses with oxaloacetate. Krebs cycle starts when acetyl-CoA combines with oxaloacetate. Pyruvate to acetyl-CoA is not part of the Krebs cycle. It's on here to just show the conversion. So if I say draw a Krebs cycle, don't put that bridge reaction unless you indicate it is a bridge reaction. It's not part of Krebs. Right? Bridge is pyruvate to acetyl-CoA. Did you guys call it that? Acetyl-CoA, we're saying, has 2 carbons from the acetate. Right? Acetyl-CoA is just acetate plus coenzyme A. Have you ever seen the structure of coenzyme A? It's relatively large. There's probably 15, 17 carbons in there. It's got a ring structure. It's a coenzyme. Enzymes use that to hold on to molecules. So coenzyme A is attached to lots of different molecules in the cells, including acetate. But when it's attached to acetate, we call it acetyl-CoA. So essentially we have 2 carbons. The carbons in coenzyme A are not included. Have you ever done this? Track carbons through this, right? I remember when I was an undergrad, I took principles of biochemistry, I took metabolism, I took molecular biology because I knew I wanted to go to grad school. And in metabolism, the guy made us track carbons and follow them, and I just went, really? And then I went to grad school and I went, that's helpful. So we're not going to do that, but what I do want you to remember the numbers of carbons because it helps things make sense, right? So when those combine, we end up with 6-carbon citrate, right? So CoA comes off when those molecules condense. Now, essentially, we're in the first molecule of Krebs cycle here. And again, this is like it's on its island, right? It's just Krebs cycle doing this. But in actuality, the Krebs cycle is kind of the center of Grand Central Station. It's what's called the nanoplerotic pathway. It's involved in a lot of converting of molecules and also oxidation for energy production. So Krebs cycle is a central point for metabolism in cells. And you can imagine it's very important because lots of organisms use it in ETC. So, okay, citrate combines isocitrate. 6 carbons, rearrangement of the molecule. That's it. So when you draw this, all you have to do is go citrate, 6 carbons. Isocitrate, 6 carbons. When you go isocitrate to alpha ketoglutarate, you lose a carbon to CO2. That's called breathing, right? That's respiration, right? Using oxygen, generating CO2. Lost a carbon. We also pulled off electrons. Added the electrons to NAD to get NADH. NADH can go to ETC. Every time we make an NADH in Krebs cycle, it's potentially 3 ATP. Next reaction, alpha ketoglutarate converts to succinyl-CoA. Lose another carbon, that's the last one we'll lose. CO2 comes off, again, reduce NAD. 2 more electrons can go to ETC. So we've made, potentially, 6 ATP. Succinyl-CoA, now we've added CoA, converts to succinate. Sorry, we added a CoA here. Right? In this case, we make a GTP. That's an ATP equivalent. So we count this as 1 ATP. So now we're at 7. Succinate converts to fumarate. Electrons are added FAD to get FADH2. Every time FADH2 contributes to ETC, we can make 2 ATP. So now we're at 9. This is fun, right? 4 o'clock in the afternoon, counting ATP. Now we're living. Now fumarate goes to malate, rearrangement. Malate is oxidized to oxaloacetate. 2 more electrons, which can make 3 more ATP. Now we're at 12. Here's an easy way to remember this. We're going to say each rotation of the Krebs cycle can generate 12 ATP. Just commit that to memory. Each rotation of the Krebs cycle can generate 12 ATP. Each glucose molecule supports how many rotations? 2. Each glucose molecule supports 2 rotations. So that's 24 ATP. Remember our maximum number? We said 38. We're not there yet. What about that bridge reaction? What about the bridge reaction? Every time that bridge reaction is done, 3 more ATP potentially. Each glucose molecule can do that twice. Glycolysis, we know, gives us +2. What about malate aspartate shuttle? Do that twice. We're going to say Krebs and ETC. What are we missing? Bridge. That's how you get all those ATP. Again, this is different. Sorry, this is different in some cells. Some cells use FAD and they only end up with 4. That's why you get 36. But the liver uses malate aspartate shuttle. Right? That's a lot of ATP per glucose molecule. So regulation of this is very important. So we'll start talking about that. And while I have this on the board, I just want to cover this one slide and then we're done. This is the entry point of pyruvate into Krebs. So essentially this is pyruvate to acetyl-CoA. It's extremely important for a couple reasons. One is acetyl-CoA is substrate for Krebs. The other reason it's so important, it's unidirectional. So once you go pyruvate to acetyl-CoA, pyruvate doesn't go backwards, right? And here's where this reaction is. On the inner part of the mitochondrial inner membrane is a complex called the PDH complex. It's embedded on the inside of that inner membrane. And PDH is pyruvate dehydrogenase complex. It's this guy. Multi-subunit enzyme complex. Look what it's doing. It's incorporating CoA. It's a decarboxylase. It's removing CO2. It's removing electrons and adding them to NAD. So it's one complex, several different active enzymes that are going from pyruvate to acetyl-CoA. So as you can imagine, this is a very important regulatory point. And this is actually tied to ETC. ETC can basically slow down this reaction. And so this is a key regulatory point. Now, once pyruvate is transported across, if it's converted to acetyl-CoA, acetyl-CoA does not go out. But pyruvate can. Right? Pyruvate can do this. So here's the point. Let's say, for example, ETC wants to slow down Krebs cycle. ETC can slow down this reaction. That allows pyruvate to go back into the cytosol. ETC is dependent on oxygen. So as soon as we deprive a cell of oxygen, we slow ETC. That slows Krebs by slowing this PDH. And now what happens to pyruvate? It can become lactate. That's why during high-intensity exercise, lactic acid production goes up. Oxygen becomes limiting. It forces more pyruvate to become lactate instead of acetyl-CoA. Why? What's the point of running Krebs if you don't have oxygen? Okay, so we'll talk about that coming up. All good there? Okay, thank you guys. I'll convert and load these as soon as I can. And then study guides too will be coming. Anything else I forgot? Oh, study session. Exam is Thursday. When is Tuesday evening the time when a lot of you guys have class? So we can think about either, probably evening's better, right? Probably either Monday or Wednesday evening. Wednesday? You guys want to do it evening or? Oh, there is?
Yeah.
Okay, you want to do this by email? Give me some feedback on whether or not we want to do it Monday, Tuesday, or Wednesday. Yeah, I can send out, if you want me to, I can send out options, and then you guys can kind of weigh in on what's better for you. And then, of course, if during the day, if we're in office hours, I'm happy to review as well. But evenings seem to be a good time for you guys.